So today I really want to focus on what is forensic science, what is it here that we specifically do in Tasmania versus perhaps the other labs in Australia or around the world. Is it just like what we hear on TV? I think you probably know the answer to that already. And forensic biology, so how reliable are those results that we're presenting in court? If you're on the jury, has anyone been on a jury here? Yep. Have you, any of those people actually heard forensic biology results being reported? No? Maybe? How reliable are those results that we're presenting in court? And there's a number of, I suppose, media reports that come out from time to time about different cases which are criticising forensic science. Is that a fair criticism, or can you actually rely on what we've presented and perhaps things being twisted a bit there? And then I want to talk a bit about the future of DNA identifications. So clearly, being a forensic science talk, there will be some graphic content and some trigger warnings in this presentation. It's impossible to know what's going to trigger you, and this is speaking from experience. So by all means, if something is starting to upset you, please, I won't take any offence if you stand up and walk out for a bit and then come back in, and I would have moved on to the next section. So I won't take offence if you up and leave. So the word forensic actually means suitable for a court of law or public debate. We apply the word forensic to a number of fields, so not just forensic science, not just people who go to a crime scene and find the evidence, not just people who are doing DNA profiling. It also relates to a number of fields, including pathology, document examination, fire investigation, accounting, and psychology, and so on. There's a huge number of fields that pertain to the word forensic. But the role of the forensic scientist is to perform testing and to analyse those results that we get from the tests for use in the Tasmanian court. My workplace is specifically focused on forensic biology and chemistry. Of course, here in Tasmania we also do fingerprinting, we do shoe print analysis, we do ballistic work, so firearms. Those other fields are performed by Tasmania Police, although we're in tight collaboration with those practitioners. However, we're quite separate to Tasmania Police. We come under the same departmental banner, but we like to sit over to the side so that we're seen as being unbiased. And I think that's really important that it remains that way. So is it really like CSI? Has anyone watched CSI? Yep. No, definitely not. What I will say is, though, I have watched perhaps 2 episodes of CSI, and probably only partial watching of those episodes before I have to turn it off. The equipment that I've seen on that show is state-of-the-art equipment. The sort of stuff we'd love to have in our lab. But I know full well they're not using it to the full potential by the way that they perform their tests. On TV, everything's done in an instant. For us to get a DNA profile, we're looking at least 3 days. We can fast-track results through using different techniques, but perhaps a day and a half at best to get a DNA profile. Whereas they will instantaneously obtain a DNA profile from a sample of blood, for instance. It'd be a bit boring though, wouldn't it, watching a episode on TV that went for a day and a half, so I can see why they've done it. So FSST, or Forensic Science Service Tasmania, is where I work. We're at the Tasmanian Government Laboratory, and we're split into 2 sections, forensic biology and forensic chemistry. There are about 30 scientists that work at this organisation, and then we have about 5 admin staff as well. Forensic biology is split up into 2 main areas. We have the biological examiners and the DNA profiling scientists, and our work's done both at the lab and at the crime scene. There's only a small number of us, probably might be 6 now, that are authorised to do crime scene examination on one of those people. And then we have assistants that follow us as well. So our work involves both criminal and coronial work. It's probably a 75 to 25 split of those two, so 25% coronial work. So if we have a missing person, then we'll try and obtain DNA as an ante-mortem sample to try and match to the deceased if a deceased turns up. That would be our coronial type cases. With regards to criminal, we do everything from stealings and burglaries, stolen motor vehicles, all the way through to rapes and woundings and assaults and murders. So forensic chemistry up the other end of the building consists of two main groups, the forensic toxicologists and the chemical criminalistic people. Toxicology is mostly involved in coronial work, so looking at drugs in deceased, and they also do the sort of roadside samples, so alcohol in blood if you happen to get pulled over and were drinking. The chemical criminalistic scientists are more involved in the drug identification, glass characterisation and identification, and also paint comparison work. And the two don't mix. We have our chemists and our biologists. So I've pretty much covered this, but I'd like you, the other thing that the chemists do are to attend clandestine laboratories, and really they're called up to assist the police really to handle the chemicals they find at the clandestine lab, to safely collect those chemicals and also to store them for further analysis. And what I want you to pay attention to are the photos along the bottom here. I want you to note what is the main difference between the chemists and the biologists. Yeah, the white coats. That's a dead giveaway for a chemist. But you'll also notice that in biology we're wearing masks all the time. Now chemists, we usually follow a process whereby items, if they need to be both analysed for chemistry and biology, they'll come to us first. We'll wear the full protection to try and avoid our DNA getting onto an item, so that's why we're wearing masks, gloves, sometimes hair nets, depending on the case. And then it goes to the chemists and they don't really care too much about DNA. So in forensic biology, we really focus on the identification of biological materials. So blood, semen and saliva are our three most important. The fact being that they are the most concentrated in terms of their DNA content. So if we can find blood, semen or saliva, we're on a winner. We also look at identifying faeces, and up until recently we were able to identify urine, but it's become a not such reliable test these days. So they're our main four. As I mentioned, some of us will attend crime scenes. Probably the most obvious reasons that we would attend a crime scene is to look for semen in a sexual assault case. Also at assaults or woundings or murders, our main purpose is there to look at the blood staining, but also to assist Tasmania Police with the collection of their items, their trace items, because no doubt down the track we'll be looking at those as well. And then the final product of all this is to try and get a DNA profile. So we have our own DNA profiling section, and really that's all about the identification of the person. So we go from the identification of the material to the identification of the person. And these can be either from items from a crime scene or from what we call person samples or reference samples. So if you're ever involved in a crime, perhaps as a victim, the police might ask for a victim reference sample, which can be used to compare to crime scene samples. So the main role of a forensic biologist is to perform that testing to try and identify the biological material, and then the end process is to obtain a DNA profile. So in the lab, as a biologist, I mentioned we wear masks, face masks, gloves and gowns. We work on a bench that's been cleaned with bleach and ethanol to try and get rid of any residual DNA that might be there. And then we place down brown paper as well. And everything that we use in the laboratory is actually screened regularly as for background levels of DNA, if you like. So that's part of our quality assurance program, is to test our environmental areas for DNA. The items are usually received for biology in a brown paper bag. What that allows the item to do is to breathe, so it doesn't become mouldy and grow fungus and mould and all those other things that chew up DNA. So we take a photo of the outside of the package, we take a photo of the labels, and we make sure that the seal is intact and hasn't been compromised in any way. We then take the item out wearing gloves, we describe the item, we take photos of it, we look for specific areas of damage, and we look for any trace material, so hairs, fibres, dirt. And then we look for specific biological type stains, so the obvious ones are blood, sometimes semen, saliva are visible. And then we conduct our testing. Now it might be really obvious that there's blood all over an item, so we might just test one or several areas of that staining, and we have a variety of tests that can tell us that blood's present. The type of item and the type of scenario that's provided to us will dictate the type of testing that we do. So to say that we examine these things blind without any sort of context would be false. We do have some context to what we're examining. So, for instance, we know that the item has come from a scene where a sexual assault has allegedly occurred. So we'd be looking probably for semen or some other contact. Whereas in a wounding case or a GBH case, grievous bodily harm, it's likely that we're looking for blood spatter or some form of blood transfer. And the end process of that testing, of course, as I mentioned, is to take samples for DNA profiling, and then we repackage the item and we store the samples securely away in an area of the laboratory where only a number of scientists can access, and place the item back in for storage. And the items are kept indefinitely at this stage. So now I'll move a bit into the type of tests that we are able to do here in Tasmania. These tests, oddly, they come and go a bit because either we find a better test that can replace them, or we find that one of the tests isn't as reliable as we once thought it was. But these are the tests that are in use at the moment. In fact, one of them we don't use very often, but I'll go through it anyway. So at the top we have what's called the hemostix test. This is a test that your GP might use to test urine. And what they're looking for is either protein or blood in the urine. So we actually take advantage of that and use it in the forensic field. So it's gone from a medical field to a forensic field. And we simply rub the yellow pad on the end against what we call a red-brown stain. We don't know it's blood yet, so we call it a red-brown stain. And we add a drop of water and we look for a colour change like you see up the top there. So that's fairly indicative that blood's present. But we know that this test reacts with lots of other things. So we call it a screening test. So it's really important at that stage to say, okay, we think we might have blood, now we have to confirm it. It's a very quick, sensitive test, and most screening tests are. So then we move to our two confirmatory tests for blood, which are the two down the bottom. The one on your left is called hemochromogen, and we're looking for the formation of tiny little crystals there, which you can see on the right hand, or you can see in both, in fact. That's confirming the presence of blood. So now we can say we have blood, but we can't say we have human blood because it's not specific for human blood. So in a sense, it's a screening test for human blood, but it's confirmatory for blood. And then we move to the other test on the right-hand side there, which is called the hemotrace test, and this is our industry standard at the moment for the determination of human blood. So we're looking at antibody-antigen interaction, which is just like a COVID test, right? You'd all be familiar with that now. Another screening test that we use more specifically at the crime scene is luminol. This is used in pitch black darkness, so what we have is probably trace staining. I don't know if you can see it on your left-hand side photo, but there's a small area of very faint red-brown staining, but you can't really make out what it is. This is where luminol comes into its own because it actually helps us identify patterns. It helps us identify movement as well, so we use it at the crime scene for that reason. So after turning the lights off and spraying with luminol, you can see the copper there will also react with luminol, so that's again an indication that it's a screening test because it reacts with things other than blood. But we get this beautiful shoe print as well, and what we can do is measure the exact size of the shoe print. We can even look for identifying features within that print to look, things that you wouldn't see in every shoe. So we might look at a little crack in the rubber, that sort of thing. Here's another example of luminol. So again, on the left-hand side photo, you won't be able to make out much shape definition there, but after spraying with luminol, we're looking for a hammer that's been that's had blood on it and been laid onto the floor, and we can then determine the exact size of that hammer. It's not only important to show what the weapon was, but also if we find that weapon later, we can indicate to the police, give them some intelligence of what they need to find. You're looking for a hammer. Then if they do find the hammer, and this sort of case we'd end up with 25 hammers arrive at the laboratory, one of those might be the right one, but we can swab the handle, any unstained areas, and look for the person who's held the hammer, as well as any sort of staining on the hammer to show that it's in fact the victim's blood that's on it. Then we move to semen. So we have a number of tests that will allow us to detect semen. This is our screening test called acid phosphatase, which reacts with a chemical in semen. So what we do is we press down just some clean white blotting paper over the top of the item that's wet, and we transfer any potential semen that's on the item onto that paper. Then we spray the paper, we take it away, put it into a fume cabinet, spray the paper with our acid phosphatase chemical, and we're looking for a colour change, so the development of this sort of purpley pink colour. So again, this is a screening test, so it will react with other things as well, but it's our first point of call for semen. Our confirmatory test is, of course, finding spermatozoa or sperm. And this is a case of making a slide of the material on the item and concentrating it up, staining it with what we call Christmas tree stain, and it stains sperm and nucleated squamous epithelial cells, which are the other flat cells that you see there, in a particular manner. And if we see sperm, then we can say convincingly that semen is present. We have another test which is very similar to a COVID test for semen as well. This is called SG, semeninogenin, which is a component of semen, and we're looking obviously for the two bands there to indicate the semen might be present. We use alternative light sources in the laboratory and also at the crime scene to look for semen, saliva and blood. We use different wavelengths which will absorb or fluoresce different materials. So depending on the background substrate, we can use infrared to look for blood staining, and really it's not in areas where we can't see blood staining, but it's in areas where the background's difficult to make out the exact pattern that we're seeing. So in this instance, you can see after shining infrared light on the stock of the firearm, we can definitely make out the pattern that's present there. And that's when we start to make some sort of interpretation about what might have happened. Without that, we could just swab the area that we know is red-brown in staining. We can't make out any definition of the staining, but we might be able to get a DNA profile, but we can't give any more information, any more intelligence than that. So as a bit of a bunch to finish up, these are the other tests and examinations that we perform here in Tasmania. We look for saliva using what's called a fatibus test, which is looking at the breakdown of starches in saliva. So we get a sort of a nice mottled staining appearance if saliva is present on this paper. Faeces is a really elegant, really old test using Price's reagent. What this does, it reacts with component in faeces to cause an apple green fluorescence when it's put under UV light. Very simple, very straightforward test, but one of the most pleasing to actually perform because of the result that you get. We look at hairs. Now, in terms of hairs, we don't do hair comparisons here in Tasmania. That is done elsewhere interstate. So if we find a hair at a crime scene and we take a hair from a suspect, we can't really compare the two as such to say they're from the same person. What we will do though is we will take an unknown hair, we will identify it as a hair. We can say whether it's human hair or animal hair, and we look for the root on the end of the hair, which is where our DNA will be. So without the root on the end of the hair, very unlikely to get a DNA profile, although we will try. And also one of our, not really a test, but an examination that we perform is textile damage analysis. So really we can identify based on the material whether something's been cut or torn. We can look at thermal damage. We can also perform some comparison studies. So if we've got a knife that we think might have been used in a murder, we can actually use that on either the same material or a very similar material and try and mimic the cut or the tear in that material, and then we can take that to court and say, look, see, I think this weapon might have been involved. And then we move on to one of the bigger areas, I suppose, that we perform at a crime scene, and that's bloodstain pattern analysis, or BPA. Now, this is an area that's come under quite a bit of controversy over the recent years. It's used pretty much at all crime scenes where there's blood involved, because it can actually give us some useful intelligence as far as what events might have occurred, in what order they might have occurred, what weapon might have been involved, and that sort of thing. So we'll spend a few moments looking at bloodstain pattern analysis. It's defined as the examination of the location, size, shape and distribution of bloodstains. It also encompasses some underlying sciences, so the biology, physics, mathematics, all come into those. But it is quite subjective when it's in the wrong hands. So it does take a very experienced person to be able to apply bloodstain pattern analysis correctly. And we're going to investigate some of that as well. And for someone to actually have an understanding of bloodstain pattern analysis, they really need to understand what blood is and how it behaves, how it forms patterns, how it breaks up into its components. So we know that blood is a non-Newtonian fluid. Can anyone tell me what that means?
More pressure you put to it, the more resistance you get.
That's exactly right. Yep. So it's when exposed to shear forces, it behaves differently to, say, water, doesn't it? So a really good example of that is corn flour when you mix it with water. So I don't know if you've ever done this, but it's something I used to do as a kid, where you have a solution of corn flour and water. Sitting in your hand, it's just a liquid. You can move it around just like you would with water, but as soon as you apply force to it or you crunch it up, it goes hard. So blood has that property as well. So it's very different from water in that respect. And because of this property mostly, and the reason it has that property is because of all its components. When you break it down, it's got the red blood cells, white blood cells, platelets, thrombocytes, all the rest. When it's in its complete form, it behaves very predictably. The only time it doesn't behave predictably is when it separates into, say, plasma and the rest. So we have to be careful in those instances. But it's a very predictable substance, and for that reason we can actually apply some mathematical and physics to it, I suppose, and apply some calculations. So it's a very, very useful crime scene reconstruction tool in that we can tell some of the events that might have taken place. So if we have a spatter event on a wall, we can say this has come about from a number of different options. We can't really ever walk into a scene, well, you shouldn't. Anyone that does, I'd very much criticise, walk into a scene and say, oh, this is what happened. What it tells us is we can apply a number of different hypotheses to a situation, and we can say whether it supports that hypothesis or not. So it doesn't provide the who, but it does assist with the how. It doesn't tell us the how, but it will assist with the how. And it's particularly useful where we have information from a witness or a victim, if they're still alive, where they're telling the police a story about what had happened. We can say, well, actually, none of this corroborates with their story, or vice versa. And that's really where its power comes in, I think, because anything else making a swift determination about what's happened just based on the stains that are present is actually quite dangerous. So here we have 3 different examples, I suppose, of some blood staining that we might see at the scene. So top left is what we call transfer staining. So this is typical of swipes or wipes. So blood on your hand and then you make contact with a wall. So that's called a wipe. No, sorry, that's called a swipe. I have to think about this. And then we have staining that's already on the wall and you come into contact with it and move it. That's called a wipe. Swipes and wipes, otherwise we just categorise them into transfer. Then we have in the middle and also on the right hand side what appears to be a spatter pattern, also with some large projected staining down below. So this is typical of what we might see after impact. What we can do with a nice uniform spatter pattern is to actually identify an area of convergence. So we can look at each of the individual stains within that spatter pattern. We can determine their angle of impact. So that's the angle that particular spatter stain has come from. And we can then do what's called stringing. So we can do this for usually about 12 stains. We have to be very careful in the way we select these stains. They have to be upwardly travelling stains, so not curving over. So we apply a string to each of these and we pull it out from the wall based on the angle of impact, and what we end up with is an area where the strings all converge, so they all cross over each other, and that's our theoretical area of convergence where perhaps the person's head was if they've got a head wound. So we can give that sort of intelligence, and it's quite useful in court for the jury to see, it's not just a one-dimensional type vision that they've got. Now they're actually seeing within the room where the person was when they were struck. So is BPA science? Well, someone might say, yeah, well, of course it is. It relies on scientific principles. And look, I've even got a diagram to show it's scientific. Yes, that's true. It does rely on scientific principles. We use trigonometry. We use the fact that we understand the biology of blood. We use physics to know if you apply more force to blood and to break it up, you're going to get smaller drops, and they're going to travel less distance through the air because they're under the influence of air resistance. But is it somewhat subjective? Absolutely it is. In the wrong hands, this can be actually a very dangerous field. Has anyone watched, dare I say it, have to be careful, but has anyone watched The Staircase, the documentary The Staircase? There's also a drama out now, which is, I haven't quite enjoyed that as much as the documentary itself, but there's a couple that have. So this is where hired guns become very dangerous, and we don't so much see it here in Australia, but certainly in the States, they have an expert that's called on by the defence to come in, and they're paid ridiculous amounts of money to come in and basically just give a story that supports the defence. Some of the comments I've heard in relation to bloodstain pattern analysis make me cringe. They are really, really dangerous. Not taking into account any story other than the defence story, and I think once you're doing that, you're treading in very dangerous waters. So you really need to, as a bloodstain pattern expert, be able to take into consideration all the possibilities that could explain the staining that you're seeing. So a single stain, if you imagine it, a single stain, let's take a spatter stain, so a round stain or elliptical shaped stain, that can come about by many different types of events. It could be cast off a weapon that's swung through the air with blood on it, so you can imagine you get a straight or a linear series of drops on the ceiling. That's what cast off is. It could be impact spatter, so someone's been hit with a weapon, broken the blood up into a series of drops, and then that gets dispersed within the room. It could come about through coughing, if you've got blood in your airstream and then you cough it up. So just looking at one single stain is almost impossible to say how that got there, except to say it was under some sort of force and it's moved through the air. Looking at the overall pattern, so I showed you the pattern of a spatter pattern before, which has been strung. That can help you narrow down what you were actually looking for and give you some indication of the event that took place. So now it could be, probably some sort of impact as opposed to a cast-off event or a coughing event, because we're not seeing strands of saliva and air bubbles in it and that sort of thing. But we still can't say, and this is where I think people get into a lot of trouble, we still can't say, oh yeah, a baseball bat was definitely used to cause this. We just can't narrow it down like that, like they do on TV. So bloodstain pattern definitely has its place. It definitely allows us to talk about the amount of force used. Definitely allows us to talk about the number of events that have taken place within a scene. But you have to be very careful about the interpretation. And there's people that say it's not open to contextual bias. And when I say that, it's, knowing. I was going to ask if people know what contextual bias is. There's probably some that don't. Is there anyone that doesn't understand what contextual bias is? Yep. Contextual bias is a big contentious issue in forensic science, especially in the last 10 years, say, where it's actually been shown in fingerprints in a study where people were given a number of fingerprints and they were able to make certain determinations. But then if they're given some other extraneous information, they change their mind the second time around. So that's contextual bias, where you're given information and that actually changes your opinion or changes the result in some way. That can definitely happen in bloodstain pattern interpretation. So yes, it is open to contextual bias, but without any sort of context, you can't do any bloodstain pattern interpretation. You can't really just walk into a room without knowing that someone's perhaps got a head injury versus an injury on their hand. So we do need to know that sort of information. But again, it opens itself up to some issues. And as I mentioned earlier, it can be misinterpreted by an inexperienced analyst. So I think on the stairway, the analyst there had only done, I think it was 9 cases or something like that. And, that caused, it nearly caused a mistrial. In fact, it caused a retrial, I think, in that instance. So I've pretty much already talked about these, but the BPA expert must never overstate BPA evidence in court. They must provide the multiple possible explanations for a pattern or a stain that they see and consider all alternatives and hypotheses. If there's any doubt around what they're seeing, they should never commit to a single opinion. And here in Tasmania, I don't know about elsewhere in Australia, but we certainly have peer reviews done on all our determinations by a second authorised scientist. Are there any questions on BPA? It's a fairly broad topic, I know, but it's certainly fairly topical at the moment as well. Yeah, absolutely. Yeah. It actually came about, the history of BPA is quite interesting, because the very first case of BPA was where I was actually a doctor, he was investigating a case, I don't know the specifics of it, but basically they used flies to narrow down where all the bloodstains were, because they don't have all the sort of tests and the alternative light sources that we have today. So they looked to see where the flies were landing, and of course they target straight in on the bloodstains, and they were able to sort of map the movement of flies and the position of flies to determine how the bloodstains have got there. So we'll move on to DNA profiling now, and that will pretty much cover the rest of the talk. So as Eric mentioned, DNA stands for deoxyribonucleic acid. It's very much a commonplace practice now to analyse DNA. I was fortunate enough, believe it or not, to be around when polymerase chain reaction, which is the process that we use, was first implemented into laboratories. And back in those days, you would have to move a tube containing some DNA from one water bath to another, and then back again, one water bath to the next, to the next. And the reason for doing that is we're trying to grow a strand of DNA from almost no DNA. So one strand of DNA, we end up with millions of copies of DNA. That's called amplification of DNA. And it was done, as I mentioned, in water baths. Now we have machines that just do it for us. You plug it in the machine, and it heats up, ramps up, comes down in temperature, and then it goes back up again. So the good old days. So DNA is found in almost every cell in our body. Blood, semen and saliva are good sources, as I mentioned, because they're high in concentrations of cells, and of course it's within the cells where the DNA is stored. It is, however, possible to get DNA from touched items, and people talk about sweat. It's probably not so much free DNA just floating around in sweat, but rather the cells contained within the sweat. But people talk about contact DNA, touch DNA or sweat DNA. DNA, of course, is inherited from our parents, so we get half our DNA from our mother and half from our father. And that's what makes it, that and the fact that it's unique to each individual, except for identical twins, of course, that's what makes it a really powerful tool for identification purposes. It's only a very small part of our DNA that actually codes for proteins within our body, that has a coding function. Most of our DNA is actually what we call junk DNA, so it doesn't actually have a function within our body as such. And it's these areas of junk DNA that we look at as forensic scientists. So we know that they're conserved evolutionary from one generation to the next. So because they're not coding regions, they stay very fixed. But we do know that within a population there is a variation of these non-coding regions between individuals, whereby this is what we call a full DNA profile using 21 locations is what we use at the moment. So up here is our gender indicating area. I say indicating rather than determining, because we can get males appearing as females, very rarely, but sometimes, so it just indicates the gender. So what we have is two peaks. One peak is an X peak and one peak is a Y peak. Of course, this person's a male because they have an X and a Y chromosome. So a female would appear as two X peaks on top of each other, or XX. Then we have all these other areas, and you can see they're sort of grouped. Most of the locations, in fact all, I think for this one, there's 2 peaks at each location. It's fairly rare in itself. But one of these peaks would have been inherited by this person from their mother and one from the father. They're all different locations along the DNA. Sometimes we get a single peak, like we do for a female, where it's 2 X peaks on top of each other. So a single peak would just be the same DNA characteristic inherited from their mother and their father. So that's a full single source DNA profile from a person. What we do is we compare a profile that we obtain from a crime scene to a profile that we obtain from a reference sample or a person sample. So this is a sample that the police have taken. It's a swab of the inside of your mouth, called a buccal swab, which is then placed onto a special card that's impregnated with chemicals to preserve the DNA. And we can just take a tiny punch out of that card and amplify it, and we get a DNA profile. So we compare the profile from the reference sample to the profile that we get from the crime scene. Simple as that. If the 2 profiles match, we can say that it's likely to be from the same person. We never say, because we're scientists, that it's from the same person. However, the chances of it being from someone else is very, very small, and we'll talk about that in a minute. If the 2 profiles are different, even at one of those DNA characteristics, so let's say they're the same throughout the entire profile, but instead of having a 11 peak here and a 13 peak, they have an 11 peak and a 14 peak. They cannot be the source of that DNA profile. That's all it takes is one characteristic. Of course, then you'd probably be looking at a brother or sister in that instance. So if the 2 profiles match, then we can say the stain, we can either say the stain is from that person, or it's from another person within the population that has the same profile. And that's just being a scientist, that's what we do. So unlike fingerprints, where they'll get a fingerprint match and they'll say that fingerprint has come from that person, undeniably. As scientists, because fingerprints haven't quite developed into the science world yet, dare I say it, it's come under a lot of fire in recent years, especially in the last, say, 10 years. A report came out in the States talking about fingerprints not quite being a science, so they're going to have to do a bit of work there. But as a scientist, we give the option that perhaps there is someone else in our population that also has that same DNA profile. And then we calculate the chance of that happening. And the way we do this calculation is we calculate what's called a likelihood ratio. So we have our full single source profile, and we apply 2 hypotheses to this profile. We say that this profile must have come from the person it matches, say the suspect. Versus a second hypothesis, the defence hypothesis, that it came from another randomly chosen person in the population. And what we find is, for theoretically the most common profile that we could have in the Tasmanian population, the stats of that happening, of it coming from another unrelated person, is about 1 in 4 quintillion. So we're talking 10 to the 18. Now, generally a jury can't get their head around that, and it's hard enough for me to get my head around that. And this is also very conservative, because we put a lot of what I like to call fudge factors into this calculation to make sure we're right. So we have a lot of population stats coefficients that come into it to sort of make it even more conservative than the actual true number. So that's the most common profile in the Tasmanian population. If you took all the most common DNA characteristics that we see in the Tasmanian population and put them together, we get 1 in 4 quintillion. But typically a full DNA profile using the kit that we use now with our 21 areas has stats of around about 10 to the 24, so even smaller. Does anyone know what 10 to the 24 mean, what it's called? I don't. It's a lot of zeros. And of course, this is impossible to explain to a jury. So what we do is we round it down to 100 billion, which is several factors of less strength, but still 100 billion. I think most people in the population have kind of got a feel of what a billion is, and that's why we do that. You really want to say, but it's actually much smaller, but we don't. We say 100 billion. That's our default figure that we report to. We don't say this in court, but for this audience here, to put that into perspective, 10 to the 8, which is, what's that? 300 million is the chance of dying from a shark attack. And 10 to the 8 is far less than 10 to the 24. The chance of a meteor landing on your house is about 10 to the 14. So I think that would be around about 180 million, billion, in the trillions, I think, maybe 180 trillion. So you can imagine, you never sort of imagine that a meteor could ever land in your house. That's just impossible. It's never going to happen. Well, the chance of finding 2 DNA profiles that match, and they're from unrelated people, is way, way less. Does that make some sense? Yep. So it's a very powerful tool. Of course, from samples from crime scenes, so say a car that's been stolen and the suspect or the offender has handled the steering wheel, and we take a swab of the steering wheel to try and find out who it is, you're not really likely just to get the offender on that swab. You're also going to get the person who owns the car, and maybe their son, because their son sometimes drives the car as well. So what we usually would get from a crime scene, if it's not from blood, say, or semen or saliva, is what we call a mixed DNA profile. And this particular profile here, and although it's very hard to see, you'll see at some locations there's more than 2 peaks. So there's a very small minor contributor coming through here. It's a bit hard to tell actually. Oh, this peak here, for instance, it sits well outside of these 2 larger peaks. So we have a major contributor, but we also have a minor contributor coming through. And it's actually the minor contributor that we're often interested in, because the person who's stolen the car, say for 5 minutes to go down the road, they might have only left a small amount of DNA, versus you who has driven the car many, many hundreds of times. Once upon a time, up until perhaps 3 or 4 years ago, we used to do this type of separation manually. So we'd look at a profile, and based on the peak heights between the major contributor and the minor contributor, we'd separate it out using various rules. Now it's all done for us by a piece of software called StarMix, which has actually been developed by laboratories in Australia and in New Zealand. There's a similar software program in the States that was developed in the States, but it's an incredibly powerful piece of software that takes into account all the different facets of a DNA profile, peak heights, peak ratios, to separate that DNA profile into what it thinks are the different contributors within that profile, which we as humans just can't really do really accurately. We might get, 90 or 80% of the way there, but this just takes it one step further. So what we have is this horrible mixture of peaks, DNA characteristics, and basically this software says, I'm 98% sure that contributor 1 is this profile, a series of numbers and letters. And I'm 1.3% sure that the second contributor is this profile. And of course, we take this into account, how strong that number is. So 98% sure, yeah, we'll put that on the DNA database and see if we get a hit. So talking about DNA databases, we have 2 that we have access to. We have the Tasmanian DNA database, which is very much governed by the Forensic Procedures Act, and we put DNA profiles on there and match them according to that act. So it's very specific about what we can and can't do with certain DNA profiles, depending on where they come from, whether it's from a person or from a crime scene. Versus the National Criminal Investigation DNA Database, or NCID, which allows us to do interstate matching. Now, Each of the states has different rules about how their profiles are applied to this database and what matching can occur. So it was the case some years ago where New South Wales would only match with certain other states or vice versa. So there's been a lot of toing and froing there. And of course DNA is incredibly valuable for solving cases, so actually finding if we've got an unknown DNA profile and then suddenly we get a person's DNA profile in the database and we get a match, we can be fairly confident of who might have committed a crime. An excellent example of that is the rape investigation that took place from the Cornelian Bay, where we had an offender who raped a 23-year-old woman as she was running on the running track down there. Fortunately, with DNA, within about 3 days, we were able to give the police intelligence as to who it might have been. But then, of course, we have the cases, and these are probably the most satisfying, where you can actually exonerate someone from a crime. So this was the case where an elderly lady was raped, and she had very poor vision, but she was fairly convinced that it was her gardener that committed the crime. And of course, unfortunately, this person went through pretty much hell and back because the family thought he was involved, he was charged, all the rest. Turns out, once we got the DNA profile from a young 18-year-old, that in fact, it wasn't the gardener at all. It was this 18-year-old that had committed the crime. So that was very satisfying to be able to say, to be able to let this poor guy off. So where are we as far as the future of DNA profiling and DNA identifications? Well, we're already using some of these. We don't use mitochondrial DNA profiling here in Tasmania, but we do send samples interstate. Mitochondrial DNA profiling is DNA within the mitochondria, which is the powerhouse of the cell, and really it follows the female lineage. And it's very useful for degraded samples. So it has been used for things like war crimes, where you have mass graves overseas, for instance, and the samples that you're getting out of that situation are horribly degraded and decomposed. Well, mitochondrial DNA profiling is very useful in that instance. Y haplotype profiling, we do use this in Tasmania. In fact, it's becoming more and more commonplace, particularly useful for sexual assaults, where you have a mixture of female and male DNA. We're able to actually separate out the male DNA in the sense that we use Y haplotypes, so the Y chromosome, we focus in on the Y chromosome, and that DNA is actually passed through the male lineage. So it's not as powerful as conventional DNA profiling in terms of the stats that we get, but it is useful for actually separating out that female DNA from the male DNA. Another area that some labs have adopted interstate are these platforms to allow the analysis of hundreds or even thousands of DNA characteristics at once. So I put up some DNA profiles which showed 21 DNA areas that we look at, and that's pretty powerful. I mean, you saw the stats, they were sort of 10 to the 24 type stats. These microarrays and also was initially called next generation sequencing, it's now called massively parallel sequencing, allows the analysis of thousands of different DNA areas at once. Basically, it's what we call DNA on a chip, so it is literally a tiny little chip, and chemicals are passed through and are allowed to bind to this chip, and it shows us the presence or absence of certain DNA characteristics. So incredibly powerful, but you can imagine that the analysis is also very time consuming and difficult. And of course, robotics are becoming commonplace in forensic laboratories these days, particularly in DNA profiling labs. We're about to purchase this piece of equipment. which is a Hamilton robot, basically allows us to liquid transfer without any human intervention at all. The benefit of that is humans can sometimes make a mistake. It'd be silly to say that we never make a mistake, but fortunately through our quality assurance process we'll pick up those mistakes. But robotics won't make a mistake, and if it does make a mistake, it's logged in a computer log. And then we move to phenotyping, which is another new area in DNA profiling, one that we haven't gone down the path of yet, but basically it allows us to look at appearance traits or the phenotype of a DNA genotype. And this is where we can use commercially available kits, which are available now, to look at the eye colour, the hair colour, skin pigmentation, and also male pattern baldness in a DNA profile from an unknown person. So if we can tell the police some sort of intelligence about this person that committed this crime has blue eyes and brown hair, for instance, that could be useful. Of course, there's problems associated with that in that people can wear contact lenses, people can change the colour of their hair, all sorts of things like that. There's also ancestry markers. Now we know a bit about ancestry markers with the formation of these genealogy databases that are becoming fairly commonplace now, where you can submit a sample to a company and they process it in much the same way that we would, and then they can link your ancestry and try and track down your family members, that sort of thing. Everyone's familiar with those, aren't they? Yep. The problem with ancestry markers on their own is they're not terribly reliable. They'll give you some percentage of your, I've got, X percentage African versus X percentage, that doesn't really necessarily reflect your appearance. And I think we all understand that. And then we move to this big, scary area of familial matching using these genealogy databases. This is a real scary area because it just opens up a can of worms as far as legal and ethical issues. So these databases, as I mentioned, like Ancestry.com, that's a common one that we'd be familiar with, they're set up to trace family trees and find ancestors. It's a legal nightmare because it's basically solving crime at the cost of privacy. Now, it depends who you ask, but a lot of people would say, yeah, but they're using my DNA and they're using it in a way that I never agreed for them to use it. But who owns your DNA? Certainly when it's within you, you own it. But once you give your DNA to a public database, is it still yours? So these are the sort of questions that we're having to answer at the moment. It's not commonplace, in fact it's not used in Australia yet. Familial matching is, so we will use familial matching, but not using these public databases. It's certainly used in the States, where the first known case that was solved using it was the finding of the Golden State Killer. Is anyone familiar with the Golden State Killer? So this Golden State Killer was a serial burglar, rapist and killer. They believed to have committed at least 13 murders, 50 rapes and 120 burglaries between 1974 and 1986. What the law enforcement agencies went about doing was to collect samples from the crime scenes, just like we do. And instead of just using it on their local database, like I've talked about doing, they've actually uploaded one of those profiles from this unknown offender into one of these public databases. So they've given it a name, Joe Bloggs. This is Joe Bloggs' profile. I'm giving you my profile. I'm Joe Bloggs. Please put it on your database. And because I'm interested in finding out who my ancestors are. Well, that's not exactly what they were doing. So they made this fake personal record, and in fact what they got was a partial match to a third cousin of this man named Joseph D'Angelo. That's incredible intelligence. So if you get to that point, then you can start tracking down other family members, and of course that's what they did. and D'Angelo was later charged in 2018, so quite some time had passed, which meant that some of the statutes of limitation had also passed. So instead of being charged for all these murders, rapes and burglaries, they could only charge him with a certain number of them, but it was still significant. So that's how this database has been used. And of course you can imagine the ethics around this and the debates that are taking place at the moment. And our legislation in Tasmania is actually silent on this. So there's nothing stopping us doing it, but ethically it doesn't quite feel right. So we need to investigate how we're going to go about this sort of thing. Now this is really the area that I really wanted to talk about, because I don't get to talk about this to any other groups, but I thought this is the perfect group to talk about it. Everyone will be familiar with Susan Neal-Fraser and the case that occurred there. Yeah, I am too. So there's been a lot of criticism come about forensic science with regards to that case, and there's been a lot of criticism with other cases like Chamberlain, of course, and rightly so. There was good results came out of Chamberlain, in that we've changed a lot of things that we do because of that case, and it had to happen. Susan Neal-Fraser is a different matter. It's not, I have to be careful what I say, of course, but it has been through the court. It's not a forensic case whatsoever. It's been made out to be a forensic case, but in all honesty, it's not. It's very much a case of circumstance, and forensics really didn't have any play in it whatsoever. The only time it has is when we examined a stain on the yacht. We still don't know exactly what it was, but we believe it could be saliva, could be blood. But it came up, it showed up positive with luminol, which is our screening test for blood. Now saliva will sometimes do that because there's blood cells in saliva sometimes. But regardless of what it is and how it got there, we got a match to, at the time, a homeless individual by the name of Megan Vass. And that became great, a great source for the defence to use, but we haven't helped the prosecution in any way whatsoever. We've just opened this can of worms basically, but we have to report what we find, so we've got to match to a homeless person at the time. And she has no memory apparently of where she was at the time and how that DNA got there. Unfortunately, not so much the media, because the media are actually very good in reporting forensic science reports, but support groups and things like that have come out and put a lot of doubt, I think, into the Tasmanian population about the reliability of forensic science, and I think that's really unfair. And so I'm voicing that today. So I'm going to talk about why you can rely on the forensics results that we present in court. Firstly and foremost, and probably the most important thing, is that we're a member of the National Association of Testing Authorities in Australia, NATA. I'm sure there's people here who are familiar with NATA. It covers a lot of industries, and basically this is an external body that comes into our workplace and audits the workplace. They make sure that we're adhering to certain standards, very tight standards in fact, under this ISO 17025 standard. Under these standards, we have to have a quality assurance program. And what this involves is ensuring that our training's up to date and adequate, that the people performing the tests, all the tests I've talked about, are authorised to do so. And by being authorised, you have to show your ongoing proficiency. So probably a week, a year, I spend doing authorisation tests where I'm not doing casework. And it seems like such a waste of time. But if I don't do that proficiency tests and show that I'm actually capable of doing these tests and getting the right results, then really my work's useless, I think. So this is just an example of a Bloodstain Pattern Analyst proficiency test that we might get once a year. So on the left here we've got individual spatter stains, and we have to determine the angle of impact. And of course the people who have created this, the external agency who's created this test, they know exactly what angle they've applied these stains at, and we have to get within 3% or we're not proficient. Then we might get a pattern, and they will ask us, well, what's the predominant pattern that you're seeing here? And in this instance, it's impact. And then we get, there's been a stain on the floor and someone's stamped on it, that sort of thing. So it's an impact pattern. And the same with this one. What's the predominant pattern here? It's a transfer pattern. And then we get one at the end, which is really complicated, and we have to give a paragraph description about what we're seeing. And this is where we start talking about sequencing. So stains, projected stains were deposited on the surface. They've run down, and then sometime later, there's been a wipe through that staining, that sort of thing. So we have to demonstrate to these testing bodies that we're actually proficient at making the determinations that we do. Also, part of this quality assurance program is document control, where we all have to adhere to our testing. We're all following the same procedures. So if there's any deviation from our procedures, it will be noted. And our methods do change from time to time, but we have to ensure that everyone is following the same procedures. So that comes into document control. We perform quality control. So all our reagents are actually tested before we put them into use to make sure they're free of DNA, for instance. And we have negative and positive, probably one of the most important things really is the fact that we have negative and positive controls in all our tests at the relevant stages of testing. So we have to ensure within a batch of DNA profiles, we run a positive control, which actually we run two. We run what's called a cell line. We have to make sure that we get the right DNA profile from that cell line. And we also run a staff sample, one of the chemists, that they provide their DNA, and we have to ensure that their profile is correct as well before we can actually pass the rest of the DNA profiles within that batch. And all the test results are read by a second authorised scientist, so this is someone else who's also authorised to perform that testing, and they do that blind. So I'll type a batch of DNA profiles, or I'll read a human blood test, and then I'll go away, a second person will come in and read it without me being present. And then all our opinions and our reports are also reviewed by a second authorised officer. And if that's not enough, then it's up for assessment by the court, the judicial process. So the lawyers, the prosecutors, defence, the judge or magistrate can also question the reliability of those results and ask questions of us about our opinion. So it's really important that we provide impartial evidence, and we'll only provide an opinion if we're qualified to do so. And at the end of the day, it's the jury or the magistrate that decides how much weight they're going to place on our evidence and whether they trust us or not, I suppose. What about the media reports? Are they accurate? Well, actually, I've never really had a problem with any of the media reports. They've all been pretty accurate, and they will essentially just report the facts. Unfortunately, it's a statement from, say, support groups and things like that. They make it very unfair, I think, in some instances, where they'll twist the truth a bit. So that's the only instances where I've disagreed. But something I want you to think about, and a lot of research has gone into this and continues to go into it, is the fact that I'm just present in front of the jury giving my evidence. The fact that I've been called up and I appear on the stand, is that enough to sway the jury? We have this effect that's now called the CSI effect, believe it or not, where if you get a scientist, and especially if you dress them up in a white lab coat, and you put them in front of a jury, it just makes such a powerful statement in front of a jury that that could have a serious effect. And so that effect's being researched quite closely at the moment. So you have statements like, well, she's an expert, so they must be saying the right thing. Or the prosecution have called this guy in, so they must have something against him. They must have some evidence on him. Therefore, he's got to be guilty. That's without even hearing the evidence. So I think this has got to be taken pretty seriously in the future, and perhaps there's ways around this, but I don't quite know the answer to it. But whether it's the expert not appearing in front of the jury, but somehow it's being presented to the jury in a non-biased way. But yeah, something to think about. So that pretty much finishes my presentation, and I'm more than happy to take questions at this point. I will say though that there's probably some things I can't answer. It just depends whether it's been reported or not and whether it's gone through the judicial process. Probably up, oh, we're just waiting on a microphone.
What's the career path for a person entering this field of science, and are there different ways that this might happen?
Absolutely. So up until probably 15 years ago, there wasn't such a thing as a forensic science degree, but nowadays you can go straight into university and study forensic science. I don't know if it's a problem or not, but I see it's could be a bit of an issue in that in these courses you get exposed to a huge variety of different areas of forensic science, rather than just focusing on, say, forensic biology or forensic chemistry. So, so what we tend to do is choose people who have got a really well-balanced and rounded science background. You'll certainly find at our workplace we have people like myself who have come from a medical science background. We have people who have got a statistics background. One employee's did a Doctor of Philosophy in forestry. Another person studied seals in Antarctica. So you can see it's that real thinking outside the square type approach that we're looking for. But certainly a science degree, and most of our employees would have honours these days, and in fact half of us have got a PhD.
Excuse me, mate. How many prisoners would be enlisted for your work? I'm not sure whether you're allowed to say it or not.
I'd be allowed to say it, but I don't know the answer. Through our work specifically?
Yes.
Yeah, that's a really, really good question, actually. I don't know the specific number, but I'd say very few are actually in there because of specifically what we've done, like maybe 10%.
Oh, thank you.
We're really... There's a section of the lab that really focuses on the major crime, so crimes against the person, murders, assaults, rapes. They're the ones that DNA really has an impact on because often it's an unknown person and we can put a name to it. It's very rare that a case will actually, or someone will be prosecuted based on DNA alone because it has to be tied with other things generally. Because DNA can be transferred, so we need to take into account, just because there's a DNA profile there, how did it get there? That's the important thing. It might have been transferred there, by someone else that picked up a cigarette butt and dropped it in the crime scene, for instance. So it's actually, although we provide a lot of DNA matches to the police, it's only a certain proportion of those that will be followed through in the court process because they haven't got other information that they can go on as well. Does that help? Yep.
Because it's a time-consuming, I presume, expensive process, how minor would a crime be before it's not actually considered feasible?
As long as it comes under the Forensic Procedures Act, we'll do it. Yep. That includes wildlife offences now, fortunately. There was a time when we couldn't do those, but it doesn't have to have any monetary value to it. It can... Yeah, as long as it comes under the Act, and probably the smallest of crimes would be burglary or stealing is what we deal with. Yep.
Who actually pays for the work of the forensic laboratory?
So the
Are you independent? You're not sort of like the prosecutor wouldn't have to pay for your information or the defence or vice versa?
That's correct. So the Tasmanian government, we come under the Department of Police, Fire and Emergency Management, although we're not police officers. So we sit off to the side, but we still come under that banner, and it always scares me a bit. But, but essentially all our work is paid for by the taxpayer. We do defence cases. I don't know the ins and outs of who pays for that, but I assume Well, I know that in some instances the defence would pay for that work to be done.
There has been some publicity recently about some problems in forensics in Brisbane. Part of it, I think, was that many samples were dismissed as being inadequate for DNA analysis. I'm not sure if there are other problems too in inaccuracy. Could you comment on that?
I can comment on it. As far as what's been reported, this is a massive one for forensic science. It's very much in the early days, and an inquiry has been opened into this. So Queensland go about dismissing some of their samples, I suppose. They use a rule whereby if they don't get it up to a certain amount of DNA in their sample, they won't go ahead and amplify it and try and get a DNA profile from it. I think that's a fairly standard approach across the country. However, their cut-off might be different to everyone else's. But also, I think where they're coming under the most fire is that they haven't been amplifying the good sources of DNA. So blood is the one that they've mentioned in the report. And all I can really say is that if we had a blood sample and it was giving the right amount of DNA in our quant system, so we've measured how much DNA is there, then we would expect a DNA profile. Now that's one of the issues that have come up, is that they didn't get a DNA profile from a blood sample. Of course, just based on the information that they're giving us, we have to assume that they have in fact tested it and found it to be blood, or are they just querying whether it's blood? But certainly if we've tested a sample and we've found blood, we would expect a DNA profile from it. The other issue is that anything that we consider might have blood, semen or saliva in it, so our good sources of DNA, we would amp regardless of how much DNA we measure. So that's where we're quite different from that lab. But that's about all I can comment on.
If someone's had a bone marrow transplant, how does that affect their DNA? And has that ever been a factor in any investigations that you've been involved with?
It's definitely not been a factor in anything I've been involved in or in my time at the lab. But yes, it can affect your DNA profile in that you're receiving a sample from someone else usually, or not usually, but sometimes, so a blood transfusion. Usually a bone marrow transplant would be your own bone marrow that you get back, so that wouldn't affect you. But after a blood transfusion, you will have a proportion of your blood will contain someone else's blood, and therefore someone else's DNA. So there will be a period of time where if we took a sample, we would probably get a mixed DNA profile from you, which is a bit weird. But yeah, so it definitely could affect the DNA. It's only a transient period though, it's only a short time.
The spatter patterns and strings looked a bit quaint. Don't you use droplet tracing computer modelling or anything like that?
It can be done, but we actually find that the jury responds much better to seeing a physical stringing. There's certainly software programs available. We haven't used them, and to use them would be actually harder to do. But no, we just don't use it because we found that the jury responds really well to the stringing. There's other theoretical calculations that you can perform as well, which will give you a number, but the jury it's really hard to fathom what that number means, but if we actually show them physically what it means, then it's a much better portrayal of the results, I think.
Have you done this lecture for juries?
Not for juries. Well, if we've done it for juries, we've done it inadvertently because we don't know who they are or whether they're going to be a juror in the future. Perhaps if you now become a juror, you'll have some better knowledge about it and we'll have a better way of assessing what we're saying in court. We've certainly given presentations to prosecutors, to defence, legal aid, magistrates and judges. So we try and keep them educated as much as possible, because as you can imagine, it's a developing field, and we're always coming by new tests and technologies, so we try and keep them up to date as much as possible.
Thank you.
Apart from watching bad television, many of us here in the north became interested in this area when there was a case where the blood sample was collected before DNA testing was available. The collector was a leech.
Yes,
You can remember the case.
I do. It's amazing police work. Yeah.
How much effort is put into a retrospective case like that, or how does it arise?
So we have a cold case unit in Tasmania Police who are asking us to do retrospective samples all the time. So any unsolved case, you can be rest assured that we're still working on them. As technology evolves, we're getting better kits that show up more and more DNA characteristics, for instance. So some of those older cases we've gone back and tested with the new kits. But yeah, the leech story was amazing. It was a police officer who had the foresight to think, one day we might be able to get something from this. So they've actually collected the leech, which has been sucking on the blood. So fantastic stuff. Yeah.
Is it only cold cases that you look back at, or there are people that are in jail for a long time that you can retest? Do people retest their DNA?
To be honest, the requests for that are very, very limited. We just don't get the requests to do it, but we would do it. It would come through the judicial process though, so they would raise, well, especially under the new ruling where if there's fresh and compelling evidence, you can have a retrial or a new trial. Certainly in that instance, then we would do retesting, or the testing would be done interstate. Yep.
You said that you often do presentations to students or young people. Have you any reflections or observations on how that goes across?
Usually there's one or two really keen people in it, and you can tell that they're the scientists of the future, and certainly based on the questions they ask. Some of them are completely put off by the idea that they have to deal with dead people, analyse semen and saliva, and I don't blame them really. But yeah, it's a real mixed response actually, yes.
Do you think a miscarriage of justice like happened with the Chamberlains would happen again these days, or what's improved if not?
If it was purely based on bloodstain pattern interpretation, it could happen, absolutely. Fortunately, our Director of Public Prosecutions section, they're very good on having to have multiple things that come together to form a case that they can prosecute. So I think a case based on one test result alone, that would never happen. We've learnt so much from Chamberlain in the fact that we now include positive and negative controls in everything. We get a second scientist to agree with or disagree sometimes with a result, and things are done blind where they weren't done back then. I'd never say never, but it's much less likely to happen these days than in the past. Yeah. That's very kind. Thank you very much. Appreciate that. Yeah.